In this video, we're going to take a look at DNA replication. The reason why we see DNA replication is we need to have two copies of the DNA before our cell divides, so that way each new cell will have an identical copy of this genetic material. A couple things that we need to know about DNA. It is double-stranded, or it has two strands, and it forms a double helix. When we're looking at this strand of DNA, if we just go ahead and draw one out here quickly, we know that it's going to be made up of 4 bases. And these bases that we see in DNA are going to include A, T, C, and G. A stands for adenine, T thymine, C cytosine, and G guanine. And this is what we are going to see forming bonds with one another to give us our double strand. We know that A and T are always going to bond together, and C and G will always bond together. Now, the way that I told you guys in class that we can remember this is apples in the tree, A pairs with T, carrots in the ground, C pairs with G. Now, if you are not someone who works well with mnemonic devices, if we just take a look at the shape of our letters, A and T both have nice straight lines to them, so they're going to pair together because they're similar. And then C and G have curves to their letters, so because they're similar, they're going to pair together. Whichever way works for you, to know that A and T will always pair, C and G will always pair. Now, to make our copy of DNA, first thing that we have to do is we actually have to separate this double strand to be able to make a new copy. And we have a couple different enzymes that are going to help us. Now, the enzymes that I want you guys to know is helicase is going to come in, and it's actually going to cause or separate our strand of DNA. It's going to, what I like to say, unzip your genes and separate them so we have 2 strands that we have access to. When we have these 2 strands, if we just kind of straighten it out, if we have A, T, C, G, C, T, T, A, C, and I'm just making these letters up so we can see what this looks like. On one side, our second side of DNA, the right letters there, is going to be exposed, and then we are going to see the complementary base pair is going to be on the opposite side of this DNA strand. Because we know A and T pair together, if we see an A on this side, we know there will be a T on this side. T still pairs with A, so we have a T on one strand, we'll have an A on the other. If we have a C up here, that means we have a G down here, and vice versa. If we have a G, we will see a C. C will bond with G, T to A, T to A to T, and C to G. Now, this is basically the strand of DNA that we have separated with helicase. To make 2 new strands, we're going to use an enzyme called polymerase, and it is going to read this half a strand of DNA, and it's going to be able to add the complementary base and make our new strand of DNA. I'm going to use a different color here to show you that this is a new strand of DNA that we're going to be making. We see that C will bond with G, so we will have this pair. A will bond with T, T to A, T to A, C to G, G to C, C to G, T to A, and A to T. It's basically the identical copy down here, but now we have an identical copy of what we originally had. If we go ahead and finish this out and write the identical copy over here, G to C, A, T, T, C, G, C, T, A. We will now have then 2 identical copies of our DNA, and our cell will then be ready to divide, and each new cell will get a copy of the parent DNA. Now, we like to call this semi-conservative Because if we see from our original strand, each new copy will have half of the original strand.